Welcome to part 18 of this video on the immune system. Okay, so we've now completed the cell-mediated adaptive immune response. We're now going to talk about the humoral adaptive immune response, which is initiated to pathogens which are extracellular. But remember, most intracellular pathogens, they will be extracellular for some period, and therefore often intracellular pathogens end up with not just a cell-mediated adaptive immune response being initiated against them, but also a humoral adaptive immune response being initiated against them. Okay, so let's go through what we've seen so far of the humoral adaptive immune response. So remember, it begins with antigen presentation. So here we are. Here's our antigen presenting cell, and remember, antigen fragments from extracellular pathogens are going to be mounted on MHC class II protein complexes. So you'll produce an MHC class II protein complex. It'll assemble in the membrane of the rough endoplasmic reticulum. What you'll do temporarily is you'll sit an invariant chain protein in the peptide binding groove of the MHC class II protein complex, and this is to prevent any peptide fragment binding in there before we've actually got to the appropriate place. So where is the appropriate place? Well, the antigen presenting cell will phagocytose one of these extracellular pathogens, and the lysosomes will fuse with the phagosome to break down the proteins of the pathogen into little fragments. So we've got a phagolysosome here. The MHC class II protein complex with the invariant chain in its peptide binding groove will then move through the classical secretory pathway through the Golgi apparatus into a secretory vesicle, and then these secretory vesicles will fuse with the phagolysosome, and in the phagolysosome the invariant chain will be broken down and will be removed from the peptide binding groove of the MHC class II, and fragments of antigens from the pathogen will then replace the invariant chain in the peptide binding groove. So you'll now get MHC class II complexes with peptide fragments being produced in the phagolysosome here. And now what will happen is these phagolysosomes will fuse with the plasma membrane, and the MHC class II peptide fragment complexes will now be on the surface of the antigen presenting cell. And remember, for a single pathogen, it's going to have a huge number of different antigens. Each of these antigens will be broken down into loads of fragments, so potentially this antigen presenting cell is going to have a huge number of MHC class II peptide fragment complexes on its surface, with where the peptide fragments are all different. What's now going to happen is the antigen presenting cell is going to move via the lymphatic system to a lymph node. So it's going to move through the lymphatic drainage of the affected tissue to a lymph node, and it's going to, in the lymph node, look for T cells of the CD4 positive type, the helper T cell type, which it can activate. Okay, so let's say it finds one of these correct T cell clones that has a T cell receptor on its surface which can recognize an MHC class II complex with one of the peptide fragments from this pathogen. Then what's going to happen is it's going to activate that T cell in the way that we've seen. Of course, it will put B7 molecules on its surface because it has seen a pathogen, and what will then happen is you'll get clonal expansion of the population of CD4 positive T cells, so you'll get loads of these T cells produced with that T cell receptor that can recognize the MHC class II protein complex with that peptide fragment in. And these T cells are now going to differentiate into both effector T cells and also memory T cells. So most of them will become effector T helper cells now, and a few will become memory T helper cells. We now want to study these effector T helper cells and what they're actually going to do. Okay, so the humoral adaptive immune response. So now let's study an effector T helper cell. So the first thing that I want to discuss is that these effector T helper cells are going to put on their surface a molecule known as CD40 ligand. So this is going to be a molecule called CD40L, and the L stands for ligand here, so it's going to be a molecule capable of binding to another protein that's an integral membrane protein known as CD40, and we will see CD40 later on. For now, all I want you to know about CD40 ligand is that it's a molecule that's absolutely essential for activating B cells. So these effector helper T cells, their main role is going to be to activate B cells, and therefore they absolutely all need to have CD40 ligand on their surface, because this is the essential molecule for activating B lymphocytes. So I'll just label this cell up. So this is an effector helper T cell, so TH cell here, and I'll just colour it in yellow. Okay, right. The next thing that I want to discuss is that there are different types of effector helper T cells, and that they all have slightly different functions on the side of helping with the activation of B cells. So the first thing to say, these effector helper T cells, they are part of the humoral adaptive immune response, and the major thing that the humoral adaptive immune response achieves is the production of antibodies by B cells. And the helper T cells, the reason they have their name is because they help B cell activation, they help B cells. So the major function of effector helper T cells is to help with the activation of B cells, and we will be studying that process in a huge amount of detail later on. But for now, what I want to talk about is the fact that there are different types of effector helper T cell. So when the helper T cells differentiate from being naive helper T cells to being effector helper T cells, there are different types of effector helper T cell that they can become. Now, all of them put this CD40 ligand on their surface once they become an effector helper T cell, and that's the molecule that allows them to help with the activation of B cells. But they're all going to have other functions on the side of activating B cells, and it's these other functions that differ for the different types of effector helper T cell. So let me now give you the different names of the effector helper T cells. So the major types are the ones that I'm going to give you. There are other types beyond what I'm going to tell you. I'm just going to tell you the major types. I'm not going to go right to the cutting edge of research. I'm just going to focus on the major types. So here come the major types then. So there are the T helper 1 type effector helper T cells. There are the T helper 2 type. There is the T helper 17 type. And there is also what's known as the T follicular helper T cells. So these are, this stands for follicular helper. And the follicular refers to the B cell follicles inside of lymph nodes. So follicular helper T cells, we would refer to these ones as. Okay, so these are the 4 major types of effector T cells, effector helper T cells that I'm going to talk about. As I say, if you read the current research literature, you will find out about other types of effector helper T cells. I'm not going to mention them. So things like, for instance, regulatory T cells, I'm not going to mention them because the picture still isn't that complete about what they actually do, and therefore I'm just going to stick with things that are well understood at the moment. There's lots of parts of the immune system that I haven't mentioned in this video. I mean, there's a whole topic called gamma delta T cells, where the T cell receptor, instead of being made up out of an alpha chain and a beta chain, is made out of a gamma and a delta chain, which I haven't touched because it's not very well understood at present. So I'm just going to focus on things that are well understood and which make sense, rather than the cutting edge level of immunology. Okay, so here are the 4 types of effector helper T cells that I think it's important to know about. So what I want to now talk about is what are their slightly different functions, because they all do have slightly different other functions on the side of helping with the activation of B cells. So the first thing I want to say is it's not particularly well understood what determines the ratio of production of these different types of effector helper T cells. So in different adaptive immune responses, in different humoral adaptive immune responses specifically, you'll get different ratios of these effector helper T cell types being produced. So in some you might get a huge number of the effector helper T cells being the T helper 1 type, whereas in others you might get loads being the T helper 2 type and not very many being the T helper 1 type. So the ratio of production of these different types of helper effector T cells is different in different humoral adaptive immune responses, and it's not well understood what determines the ratio that you produce these different types of helper effector T cells in. So, let me now talk about what the functions then of these different effector helper T cells is. So T helper 2 and T follicular helper I'm going to start off with. So these two here, so T helper 2 and T follicular helper, these two are both absolutely brilliant at B cell activation. Now, all of the different types of effector helper T cells that I've got listed here, T helper 1, T helper 2, T helper 17, T follicular helper, they are all capable of helping B cells become activated. However, T helper 2 and T follicular helper are absolutely brilliant at it. Okay, so they're the special ones that are brilliant at it. They're better at it than the T helper 2 and the T helper 1 and the T helper 17 are. And one of the major reasons for this is that they secrete a molecule known as interleukin 4. They produce a lot of a cytokine known as interleukin 4, which helps B cells to activate, whereas T helper 1 and T helper 17, they don't produce that much interleukin 4. So T helper 2 and T follicular helper, they're the major two which are involved in B cell activation. T helper 1 and T helper 17, they can help B cells activate because they do have CD40 ligand, which is the essential one. As soon as you become an effector helper T cell, you express CD40 ligand. The naive T helper cells didn't have that on, and that's the thing that allows you to help B cells activate, but they're not as good at it as the T helper 2 and the T follicular helper are. Okay, so next what I want to talk about is the function of the T helper 1 cells. So let's go to T helper 1 next. So T helper 1, as I say, they can help B cells activate, and that is one of their functions, and that's the major part of the humoral adaptive immune response. But they also have another function, and their other function is the production of a cytokine known as interferon gamma. So IFN gamma for short. So this stands for inter, that's the I, and then feron, that's the F and N. So interferon gamma. Okay, so what does interferon gamma do? Well, interferon gamma is very good at activating macrophages. So let me discuss this in a little bit more detail. So if you produce loads of T helper 1 cells, what can happen is these T helper 1 cells can get into the bloodstream and they can be recruited to the site of infection. So we've been through how they will get into the bloodstream. They'll continually circulate between the lymphatic system and the blood, so they'll get into the bloodstream. They will also be recruited by the microvasculature at the site of an infection, because the microvasculature has undergone the inflammatory response and has the correct molecules on the surface of its endothelial cells to recruit these cells into the bloodstream. And the recruitment will be exactly as we said for cytotoxic T cells, i.e. it will involve sialyl Lewis X on the surface of the T helper 1 cells interacting with P and E selectin to perform the rolling stage, and then you'll have VCAM1 on the endothelial cells and VLA4 on the surface of the lymphocytes to perform the tight adhesion stage, and then diapedesis will follow and migration. Okay, so T helper 1 cells can go to the site of infection, and when they get to the site of infection they'll start producing interferon gamma. Now what does the interferon gamma actually do? Well, it acts on macrophages and it gives the macrophages new powers. So I always, when talking about interferon gamma, think of a part of Harry Potter 4. So Harry Potter and the Goblet of Fire. Now in Harry Potter 4 there's a chapter where Harry goes to visit Sirius in a cave, sorry, I almost said cage, in a cave, and he takes him some food, and they're having a chat, and they're talking about Barty Crouch, the high up one in the Ministry of Magic, and they're talking about how Barty Crouch gave the auras new powers. So in the previous time when Voldemort was rising to power, and then Voldemort was beaten by Harry and everything started falling, Barty Crouch gave the auras new powers in that last time, and he gave them the powers to kill rather than just to capture. And I kind of think that that's analogous to what the T helper 1 cells do to macrophages. Interferon gamma is the permission. It's a signal to the macrophages saying, you have my permission to use absolutely brutal mechanisms to deal with this pathogen. It gives the macrophages the permission to use more deadly weapons than they were using previously. Just like Barty Crouch gave the auras the permission to kill rather than just to capture. That's what interferon gamma effectively does. So T helper 1 cells, they tend to be produced when macrophages are having a problem with phagocytosing bacteria. So there are certain bacterial species that are extremely resistant to phagocytosis. There's a very famous one that's extremely resistant to phagocytosis, which is Mycobacterium tuberculosis. So here is my picture of a phagosome with a pathogen inside of it. And let's say that this pathogen is the pathogen that causes tuberculosis, which is known as Mycobacterium tuberculosis. Okay, now what's so horrible about this bacterium is that it resists phagocytosis incredibly well. So we can engulf it absolutely fine. We take it into a phagosome beautifully, but once we've got it in the phagosome, it has loads of mechanisms for stopping the phagocyte from actually being able to kill it. And in fact, it can live inside the phagosome perfectly happy, and it even starts breeding inside there. It starts reproducing inside of there and producing more Mycobacterium tuberculosis bacterial cells. Okay, so Mycobacterium tuberculosis is an example of a bacterium that it is really difficult to actually kill by phagocytosis. So the macrophages need, in this case, to be given the permission to use more brutal mechanisms to try and destroy this pathogen. And interferon gamma released by T helper 1 cells gives them that permission to fight fire with fire effectively and use more brutal mechanisms. So in the innate immune system, macrophages are only allowed to use some of their more tamed down tricks for phagocytosing bacteria and killing the bacteria. Once the adaptive immune system has been turned on and you've produced lots of T helper 1 cells, and you will end up having a T helper 1 preponderance, i.e. you'll produce lots of T helper 1 cells as your effector helper T cells, if there is a problem with phagocytosing the bacterium, if the bacterium is resisting, or indeed the pathogen, I should say, rather than just limiting our attention to bacteria. If the pathogen is resisting phagocytosis, then you'll tend to produce lots of T helper 1 cells. These will go to the infected site and they will produce interferon gamma, and this will give the macrophages permission to use more drastic mechanisms to kill the bacteria, or the pathogen rather. Okay, so that's another function of the T helper 1 cells alongside their function in activating B cells. Remember, all effector helper T cells are capable of activating B cells. And you'll notice that this mechanism, it doesn't really rely on the T helper 1 cell actually being specific to a certain fragment of the antigen at all. When we were talking about the cell-mediated adaptive immune response, you saw how those effector cytotoxic T cells were only capable of killing the infected host cells that had put on their surface a fragment of the antigen from the pathogen that this effector cytotoxic T cell was specifically tailored against. Whereas this, there wasn't any part of that here. The mechanism of actually taking effect was completely non-specific, didn't rely on this T helper 1 cell having a T cell receptor that was specific to the antigen of this pathogen in any way at all. It just relied on it producing interferon gamma. Okay, so these other mechanisms, you'll see that that's a running theme, that they're all quite non-specific. It doesn't rely on the specificity of these helper T cells at all. And indeed, it's the activation of B cells that relies on them having a specific T cell receptor directed against a specific fragment of a specific antigen from the pathogen. So these other functions aren't particularly specific, whereas the activation of B cells is specific, as we'll see. Okay, so that's the side function of T helper 1 cells, i.e. helping with the phagocytosis process by macrophages. And really it's just macrophages that have these enhanced weapons rather than neutrophils. So this is a macrophage over here. Okay, so let me now tell you about some of the other types of effector helper T cells. So I want to now also talk about the side function of T helper 2 cells. So I've said T helper 2 cells are absolutely fantastic at activating B cells, and they are, but they also have another side function just like the T helper 1 cells. And again, this involves the secretion of a cytokine that will then help other cells of the immune system. So T helper 2 cells. So T helper 2 cells, their side function is that they can secrete the cytokines interleukin 5 and interleukin 13. Now, what do these cytokines do? Well, they call for a cell of the immune system that we haven't actually discussed at all in this video. So I'm just going to discuss it now, which is the eosinophil. And the eosinophil is a cell type, a very powerful cell type, that can be used if you're dealing with a multicellular pathogen. So for instance, if you're dealing with a parasitic worm, macrophages find that very, very difficult because the parasitic worm is a lot bigger than a macrophage. So they struggle to attack a parasitic worm. So we have more powerful cells for attacking multicellular parasites. So these are the eosinophils. So let me introduce you then to eosinophils. So eosinophils, firstly, let me just draw a picture of an eosinophil. So an eosinophil has a bilobed nucleus. So like the neutrophils, they have a more complicated shaped nucleus than normal cells, but not quite as complex as the nucleus of a neutrophil. Their nucleus only usually has 2 lobes, like so. And the reason they're called eosinophils is that when they are stained with the dye eosin, which is a common dye used in histology, they stain bright red. So eosin generally stains things red. Eosinophils take up the eosin dye fantastically well, and therefore they end up bright red. And in fact, they are a very distinctive cell when you actually see them, because they generally look like a sunburnt face with a pair of sunglasses on. So their bilobe nucleus looks like the pair of sunglasses, and then their bright red cytoplasm looks like the sunburnt skin, of course. So a very easily distinguishable cell. So eosinophils are circulating in the bloodstream. They're produced by the bone marrow. They're circulating in the bloodstream. They are called to sites of infections by the cytokines interleukin 5 and interleukin 13. So let me now complete the story. So if, let's say, your pathogen was a multicellular pathogen, what's going to happen is you're going to have a T helper 2 preponderance in that case. So you'll produce most of your effector helper T cells as T helper 2 type effector helper T cells. And these will go into the bloodstream, at least some of them will go into the bloodstream, and then they'll be recruited to the site where the multicellular pathogen is present. And once they're actually in that site, and the recruitment process, by the way, will be exactly as we've stated before, involving the same processes of rolling and tight adhesion, involving the same molecules. And once the T helper 2 cells are present in that tissue, what they'll start producing is interleukin 5 and interleukin 13. And these 2 cytokines, they are what call for eosinophils and to come into the site of infection. So interleukin 5 and interleukin 13. So it's not particularly well understood exactly how interleukin 5 and interleukin 13 call for eosinophils. What is known, here's the fact that is known. The fact that is known is that if you raise interleukin 5 and interleukin 13 in the tissue fluid of an affected area, then you end up with loads of eosinophils being brought into that area. Exactly how that occurs, we aren't particularly sure. Something must change with the endothelial cells. So interleukin 5 and interleukin 13 must act on the endothelial cells of the capillaries and postcapillary venules of the microvasculature of the affected area, and they must cause the expression of some other molecules that are involved in the recruitment of the eosinophils. The exact molecules there aren't particularly well understood yet, but something about these molecules calls for eosinophils. So you start bringing eosinophils into the affected area. And what do eosinophils do? Well, they go to the multicellular pathogen and then they release vesicles that they have stored in their cytoplasm on top of the multicellular pathogen. So they have special vesicles here which are filled with horrible molecules, horrible proteins that are going to attack the cells of the multicellular pathogen. So to give some examples of the horrible proteins that they have inside their granules, major basic protein is one example, MBP for short. So this stands for major basic protein. So that's one example of a horrible protein inside the granules of an eosinophil. Another notable example is ECP, which stands for eosinophil cationic peptide. So eosinophil cationic peptide. So these are horrible proteins that will be released onto the surface of the multicellular pathogen and will just kill cells. They're just cytotoxic proteins, and they're very cytotoxic to our own cells. So this is why this is a very dangerous cell type, because it has these extremely dangerous molecules, and it has to have permission from the adaptive immune response before we can actually use this cell against a pathogen. So you have to have the T helper 2 response before you can actually call for eosinophils into a site, and then they can come forward, release these horrible molecules onto the multicellular pathogen, and hopefully that might have some effect. Okay, so that's the side function of T helper 2 cells calling for this new cell that I've now introduced you to, which is the eosinophil, a very powerful cell used against multicellular pathogens that macrophages are not powerful enough to clear. Okay, so that's the side function of T helper 2 cells. Now what I want to talk about is the side function of T helper 17 cells. So the side function of T helper 17 cells, they produce a cytokine known as interleukin 17. And interleukin 17 is fantastic at calling for loads and loads of neutrophils. So again, the mechanism isn't particularly well understood, but the empirical fact that we know is true is that when interleukin 17 goes up in an area, when you get loads of interleukin 17 produced in a certain area, you end up with absolutely loads of neutrophils being recruited to that area, more than you had even ever had before as part of the acute inflammatory response. You end up with absolutely loads of them called to that area. Okay, so what's the point of this? Well, neutrophils are absolutely fantastic at clearing fungal infections. So if you've got an infection that is due to a fungal pathogen, neutrophils are the cells that are best at actually clearing a fungal infection. So you tend to end up with a T helper 17 preponderance when you're dealing with a fungal infection. And as I say, the mechanisms, the exact mechanisms by which the ratio of the different types of effector T helper cells that are being produced is actually achieved isn't particularly well understood, but this is what we do understand, that when you've got a fungal infection, the major type of effector helper T cell that you're going to produce is T helper 17 cells. Of course, one of the big functions of these is that they can help with the activation of B cells, but then their side function is that again they can go into the bloodstream, they'll be recruited at the site of infection through the same mechanism that we've discussed of sialyl Lewis X interacting with P and E selectin and VLA4 interacting with VCAM1. And then once they're at that site of infection, they'll produce lots of interleukin 17. This then calls for neutrophils, and you get a huge number of neutrophils being recruited into that area, and that's a very powerful defense mechanism in the case that we are dealing with a fungal infection. So neutrophils are the best cell type for dealing with fungal infections. So, those are the side functions then of T helper 1, T helper 2, and T helper 17. The T follicular helper cells, they don't have a side function that is currently understood. Their main function, as the name suggests, is merely to help with B cell activation. Okay, So these are the side functions of these effector helper T cells. We are not going to discuss the side functions of the effector helper T cells any further. What we are going to discuss from now on is just their function in B cell activation. And as far as we're concerned, all of them are capable of activating B cells. So we're not even going to distinguish anymore between T helper 1, T helper 2, T helper 17 or T follicular helper. We're just going to call them effector helper T cells because they're all capable of helping with the activation of B cells. And this is the major part of the humoral adaptive immune response. You should see these other parts here as just a side little bit that's stuck on there, which is part of the adaptive immune response but isn't the main part of the humoral adaptive immune response. And these parts, as I've stated and stressed previously, these don't actually rely on the cells that you're producing here having this specificity, this incredible specificity to a certain fragment of a certain antigen of the pathogen. They don't use that at all. Whereas the activation of B cells is going to use the fact that they have a specific fragment of a specific antigen that they are targeted against, as we will see. You will understand how that all works later on. Okay, so we'll have a break here, and in the next video we will talk about B cell activation. But before, of course, we can get onto B cell activation, we'll have to have a big talk about B cells, just like we had a big talk about T cells. What I do want to say is that I've talked about how T helper 1, T helper 2 and T helper 17 can go into the bloodstream and then be recruited to the affected site. A lot of these helper T cells will stay in lymph nodes, however. So we know how lymphocytes can circulate between the bloodstream and lymph nodes. What's going to happen is these helper T cells are going to disseminate around all of the different lymph nodes around the body. So you may have produced all of the effector helper T cells in one lymph node in the body, the lymph node where the original naive helper T cell was actually stationed at the time when it was activated by the antigen presenting cell. There was where clonal expansion took place and then differentiation into the different types of effector helper T cells. However, what's going to happen is they're going to continue circulating between the lymphatic system and the blood, and therefore they're going to end up disseminated all over the body. And that is important. That's the reason I'm bringing it up, because the B cells might be in a different lymph node. The B cell that is going to need these effector helper T cells might be in a different lymph node, and therefore it's important that we are disseminating our effector helper T cells all over the body into the different lymph nodes all over the body, so that we have them wherever the B cells that need them are going to be. Okay, so we'll have a break here, and in the next video we will take a break from discussing the adaptive immune response, and we will talk for a long time about B cell production and the way in which B cells all gain specificity to a specific antigen.